Hi everyone, welcome back to Microbial Genetics, and we are now already in part 3. So on the first two videos, we have discussed the introduction to microbial genetics, covering the structure of nucleic acids, no? And then on part 2, we have discussed the DNA replication. Okay, so today we'll be discussing gene expression. Okay, that involves transcription and translation process. Okay, all right. So what is gene expression? Okay, remember we have on the first part of this lecture we have defined what is gene is. Okay, gene is a basic unit of heredity. Okay, so DNA molecule, diba, DNA molecule is a physical structure. Okay, it's a very long molecule that contains several functional genes. So we say that a gene coded for something. No, it codes for protein. Okay, so we are going to express these genes so that the bacteria uh, could manufacture those protein. Okay. Alright, so what is gene expression in the first place? It is a process by which the instructions in DNA are converted into a functional product, such as protein. Okay? So, kino-convert natin yung gene, no? Yung gene from the DNA to a protein. So, gagawa tayo ng protein. Again, our DNA is the blueprint. Okay? So, dyan tayo kumukuha ng information. Nakastore lahat ng information dyan. No? So, we just have to retrieve that particular information and convert it into a protein. Okay? So, gene expression is a tightly regulated process that allows a cell to respond to its changing environment. Okay? So, later, I'll show you an example, no? Para meron tayong context kung paano ba talaga nangyayari yung gene expression. Okay? So, it acts as both an on and off switch to control when proteins are made, and also volume control that increases or decreases the amount of proteins made. Okay, so again, let's define what is gene. Okay, so a gene is a specific segment of a DNA strand. See, segment lang po siya, no? Hindi siya buong DNA molecule. It contains the instructions that will enable a cell to make a gene product. Okay? So, ano naman yung genetic code? Okay? So, remember guys, um, Homo sapiens have the same DNA component, no? I mean, lahat tayo ito yung, actually, all living organisms utilize same genetic code. Okay? Lahat ng DNA molecules ng organisms have these four nitrogenous bases. Okay? Ang pinagkaiba lang, magkakaiba yung sequence. Okay, for example, just for a context, uh, a lion, no, for example, ang pagkakasunod-sunod ng nitrogenous base niya is ATGC. But in humans, it could be GTAC. Okay, or CGTA kabaliktaran. Okay, so doon lang siya nagkakatalo sa pagkakasunod-sunod ng sequence ng nitrogenous bases. Okay? So for example, all human beings have uh, the same genetic composition. Okay? Because we are in one species. Okay? But how can we be unique from each other? No? Yung sa sequence ng nitrogenous base sa DNA. Yun lang yung pinagkakaiba natin. Okay? So it says here, it is the sequence of these four bases that spell out the instructions for a particular gene product. Okay? We have two types of genes. We have constitutive genes and inducible genes. Okay? So constitutive genes, it is being expressed all the time. Uh, kahit walang changes sa environment. Okay? Automatic. So, lagi siyang ini-express. Inducible genes, on the other hand, expressed only when the gene products are needed. Okay? So, let's have some context, no, on expression. Okay? So, we say Some bacteria adapt to hostile environments. When we say hostile, it could be physical, physical modifiers. For example, too too high temperature or too low temperature of the environment. Okay, so how do bacteria adapt to that particular environment? Okay, for example, no, bacteria transform into dormant spores that can survive millions of years in extreme environments. So this is a vegetative cell. So this is a bacteria in normal environment or conducive environment, no? Nasa vegetative state siya. Okay? In here, kapag naka-encounter ng mataas na, for example, extreme environment, no? Kapag encounter ng extreme environment, itong cell na ito, it will be transformed into like this. It will undergo sporulation and will form a spore. This particular spore will protect the bacteria to that hostile environment. Okay? So sabi dito, the coating acts as an armor protecting the spore. The spores wait for the right conditions to bloom into harmful bacteria later once the environment is already conducive. Okay? So, see, merong need to transform yung bacteria to protect itself. So, anong gagawin niya para magkaroon ng coating yung sarili niya, no? So what it will do, it will express a gene coding for the coat, okay? Coding for the coat and express it, okay? And transform it into like this. Okay, nagigets niyo ba? So gene expression, happens, no? So happens every time, no? For example, for constitutive genes, no? It will express genes all the time, no? But for the genes, inducible genes, it will only be expressed when there is a need to, okay? For example, no? This is the entire DNA molecule of a bacterium, okay? So itong naka-highlight na red, ito lang yung gene, no? Again, when we say gene, it is a segment of DNA that codes for protein. Okay? So ibig sabihin itong part lang yung functional because it is the only segment that codes for something. Okay? This segment will be transcribed to an mRNA molecule. Again, mRNA is a carrier. Okay, is a messenger molecule. M stands for messenger. Messenger RNA, again, paano sila nagkakaiba sa DNA? Messenger RNA is a single-strand molecule, diba? Okay, and this mRNA will now undergo translation. Okay, protein synthesis. Translation is a protein synthesis, creating protein or manufacturing protein. Okay, so this protein now will perform its function. Okay, so kung i-apply natin yung previous slide, no, yung sa pag-create ng coating for the bacterium, no, protecting itself from harsh environment. Okay, so for example lang, ito yung gene na nagko-code for the coat. Okay, so ito yon. Ita-transcribe siya into mRNA, so the information from the gene will be transferred or relayed to the mRNA. And then this mRNA that contains the information will be translated into protein. Ano yung protein na yun? Yung coat. Okay? Yung coat, no? So, ayun, yung coat na yun will now perform its function protecting the bacterium. Okay? So, in brief, no? So, this is the gene coding for spore formation. Okay? So, SPO2R gene. Okay? So, it is a gene that codes for spore formation of Bacillus subtilis. Bacillus subtilis is a gram-positive bacterium, no? So, the protein encoded for SPO2R gene is composed of 224 amino acid residues. Again, protein is made from amino acids, okay? So, amino acid, baliktarin natin, amino acid is the building blocks of protein. So, see, protein, guys, are made from several amino acids. So, here, specifically, 224 amino acids, no, comprise the spores of Bacillus subtilis. So, ito yung mga yun. Okay? So, later, you'll see kung bakit ang daming maliliit na letter dyan, tapos merong mga single letter dito sa baba. Okay? So, makikita nyo, if you can see, no? So, these are sequence of nitrogenous bases. Right? So, ganito po yung itsura ng DNA kapag sa data na sila, no? Kapag ina-analyze na sila. So, AGAT, TACAGC, no? So, tuloy-tuloy yan. These are DNA bases and these are DNA sequence. Okay? So, DNA sequence yung mga yan. And, certain, okay? Certain, this DNA sequence code for spores of Bacillus subtilis. Okay? So, ganun yung gene expression. There is a need to express the gene. There is a need for certain structure. The structure is protein in nature, diba? So, the bacteria will now look for the gene coding for that certain structure, and then it will undergo transcription. Okay, transcription. So the information will now be transferred to mRNA. The mRNA will now undergo translation to form a protein needed by the bacteria. Okay, so paano ba yung process? So let's discuss. Okay, let's discuss the step by step the process of transcription. Okay, all right. So in transcription, okay, we are going to transfer the information coded in the DNA to the RNA molecule. Okay, so during transcription, we're going to synthesize an RNA using DNA as a template. Okay, so DNA yung template natin. Sa replication, we are producing new sets of DNA molecule, and ang template natin is still the DNA. Okay, so DNA to DNA, that's replication. DNA to RNA, that's transcription. And later, RNA to protein, that's translation. Okay, so in this process, the most essential enzyme we need is the DNA-dependent RNA polymerase. Okay, so unlike the DNA polymerase in replication that needs RNA primer, di ba? In order for it to start the synthesis of new DNA bases, DNA-dependent RNA polymerase does not need primer. Okay? And wala din siyang proofreading activity unlike the DNA polymerase. Okay? So in eukaryotes, again, we are using the prokaryotic transcription, no, to describe the process. But in eukaryotes ha, tandaan, the polymerases synthesize specific RNA molecules. For example, polymerase 1 synthesize ribosomal RNA, polymerase 2 synthesize mRNA, polymerase 3 synthesize tRNA, okay, or 5-SRRNA. So, sa RNA, sa eukaryote, since it is a complex molecule, complex cell, mas complicated din yung mga structures na involved sa process, no? Unlike sa prokaryotic cells. Okay? So again, transcription is a highly selective process that is strictly regulated. Okay? So we have what we call enhancers versus silencers. Enhancers are DNA sequences that stimulate transcription rate. Okay? It makes the transcription process faster. Okay? Silencers inhibit transcription. Okay? There are uh, times, no, that transcription should not push through. Okay? So it's a regulation, no, and it is warranted most of the time. Okay? So take a look at this one. So we have here the helical double-stranded DNA molecules. Okay, unwind. So, nakabuka na yan. Magkahiwalay na po. No? Magkahiwalay na. So, Itong molecule na to na orange, no, na heart shape, this is the RNA polymerase. Again, anong trabaho ni RNA polymerase? It synthesizes RNA nucleotides. Okay? Alright. So, ito yung gagamiting template strand ni RNA polymerase. Okay? So, again, polymerases synthesize new strands, okay, of nucleic acids in what end? In 5 to 3 end as well. Katulad ni DNA polymerase, it always synthesizes DNA molecule from 5 end to 3 end. So, ang ginagamit nating template, therefore, is 3 to 5 end. Okay? So, tingnan nyo. So, dito nag-umpisa, no? Dito nag-umpisa si RNA polymerase. Kasi meron na tayong RNA strand siya. So, pag ganyan siya. Okay? So, we use DNA template as a strand. Kita nyo? ACGCU. So, I know that this is the RNA molecule due to the presence of uracil base. Okay? Uracil is not present in DNA. Okay? So, see? Tingnan nyo dito. This A is adenine, no? Adenine. Ang sequence ng DNA dito is, dito tayo mag-start. Adenine, thymine, guanine, okay? So, sabi natin, adenine always pairs with thymine in DNA. Pero since we are synthesizing RNA molecule, wala pong thymine sa RNA molecule. But instead, it is replaced by uracil. No? That's why in RNA, adenine always partners with thymine. Okay? How about this? So, sa DNA molecule, thymine, no? Yung base, thymine siya. So, ano ang kapartner ng thymine pagdating sa RNA molecule? It's still the adenine. Okay? So, huwag kayong malilito. Si adenine lang, yung hindi adenine ng DNA, laging uracil ang kapartner niya. Okay? Hindi po si thymine. Pero si thymine ng DNA, papartneran siya ng adenine sa RNA. Okay? Pero sa DNA molecule, no, si thymine always partners with adenine talaga, no? Not uracil. Okay? So, ganun yun. And then, guanine always partners with cytosine. Cytosine always partners with guanine. And guanine here partners with cytosine. Adenine partners with uracil. And tuloy-tuloy yan. Okay? So, si RNA polymerase yung taga-supply ng bagong ribonucleotide. So, kita nyo, sa DNA, this is cytosine. Sinusuplayan siya ng guanine here in RNA. Adenine partners with uracil. Okay? Okay, so let's discuss now the step-by-step process of RNA synthesis or transcription. Okay, so we have the first step, the initiation. Okay, so the most important enzyme in RNA synthesis or transcription is the RNA polymerase. Okay, so ano yung ginagawa ni RNA polymerase during initiation? Okay, so first, it will look for DNA promoter sequences. Okay, so ano ba yung DNA promoter sequences? From the term itself, no? Promoter, no? These are the DNA sequence and base pairs, okay, where or it serves as a mark, no? It serves as a mark or an indicator kung saan makikita si transcription start site. Okay? Or TSS. So for prokaryotic cells, we have the two types, no? The negative 35 sequence. Okay, ano ba yung ibig sabihin ng negative 35? Paano siya hinahanap sa DNA sequence? Okay? So for example, this is your transcription site. Okay? And how would you know that it is the negative 35 sequence? If the sequence of the DNA bases are like this, TTGACA, no? Ayan siya, TGT, no? So the DNA polymerase would know that this is the negative 35. So bibilang siya ng 35 bases from here, from the, what you call this, from the DNA structure hanggang sa makabilang siya ng 35. So, it's until here. So, the DNA polymerase would know that this is the transcription start site. Okay? So another type of DNA promoter sequences in prokaryotes is the Pribno box, no? I read this as tataat box, no, para mas matandaan ko. But it is called Pribno box, okay? And it is about negative 10 base pairs, okay? BP is base pairs, okay? Base pairs are this one, adenine and thymine, guanine and cytosine. So base pairs yung tawag natin sa kanila. Alright? So if the DNA polymerase recognize the tataat sequence, no? Alam niya na it's already one of the DNA promoter sequences, no? So, kapag nakita niya yan, bibilang lang siya ng 10 base pairs and the DNA polymerase or what we RNA polymerase, sorry. RNA polymerase would know that it is already the transcription start site here. Okay? So again, in eukaryotic cell, magkaiba din yan, no? So sa prokaryotic cells, the Pribno box, but for eukaryotes, we have what we call the Hugness box, which is about negative 25 base pairs from the transcription start site. We have the Kaat box, which is about negative 70 to 80. Okay? So iba-iba, no? Iba-iba ng promoter sequence. But then, the RNA polymerase always make sure to look for this first para makita niya yung transcription start site. Okay? So, the DNA, again, no, it needs to be, kailangan niyang mag-unwind. Okay? Para makapag-code siya into RNA. Okay? So, unwinding of DNA occurs to create a transcription bubble. So, tingnan niyo to, ha? So, this orange one is the RNA polymerase, no? So, dito nag-start. Dito nag-start yung RNA, what we call this? RNA synthesis. Okay? So, the DNA, i-unwind siya, no? So, paghihiwalayin siya. Okay? And then, dyan papasok si RNA polymerase, no? And starts synthesizing RNA strand. Okay? Itong green is the RNA strand, no? So, itong area na to, where the RNA synthesis is happening is called the transcription bubble. Okay? That is transcription bubble. Remember guys that this strand right here is the non-template strand ng DNA, ha. So hindi yan yung kinocode ng RNA polymerase. Okay? So yung ginagamit natin is the 3 to 5 end template ng DNA molecule, no? And ang product natin is 5 to 3 end RNA. Okay? So kapag natapos ng i-code yung area na to, ni-rewind sila, okay? Tiniturn siya ulit sa helical structure. Okay? Pero habang nagpo-proceed yung RNA synthesis, no? Ina-unwind siya dito para mas madali para sa RNA polymerase mag-synthesize ng RNA. Okay? So next. Next step is elongation process, no? We already initiated the RNA synthesis, no? So take a look at this diagram. Makikita nyo, meron na, no? Nag-umpisa na yung transcription. Okay, this is the RNA strand already. Pero tuloy-tuloy pa yan, depending on the number of bases or kung gaano kahaba yung DNA, no? Na tinatranscribe ng RNA polymerase. So during elongation, nagdadagdag lang dito sa 3' ng RNA molecule ng additional nucleotides, RNA nucleotides, okay? So tuloy-tuloy lang yan, depende sa kung anong nitrogenous base yung kailangan, no? So the local unwinding of the DNA continues in the 5 to 3 direction, okay? So habang nagta-transcribe, no? Yung unwinding sa part na ito, tuloy-tuloy din. Okay? Kasi tuloy-tuloy yung pag-transcribe ng RNA polymerase. Alright? So again, nagdadagdag lang siya during elongation, nagdadagdag lang ng nucleotides para masumaba yung RNA. Okay? So for example, no, take a look at these bases. So sequence of bases in the DNA template, nakatayo lang siya, no? So for example, ang sequence is adenine, thymine, guanine, cytosine, and so on. Okay? Kung itatranscribe itong DNA template nito into mRNA molecule, magiging ganito siya. So, since adenine yung nasa DNA template, ang corresponding base niya is uracil. Okay? Since thymine yung kay DNA, ang corresponding base niya is adenine. Alright? So, since this is guanine the corresponding base is cytosine. Vice versa, no? Kung cytosine yung nasa DNA, guanine din yung nasa mRNA. Okay? So, ito ngayon yung kakalabasan ng RNA molecule mo. Remember, RNA molecule is a single strand nucleic acid. Okay? And it contains uracil rather than thymine. Okay? Right. So, let's say, natapos na yung elongation, no? Let's say, uh, na-code or na-transfer na lahat ng information or na-kopya na lahat ng information from the DNA molecule to the RNA. Okay? So, kailan siya matatapos? Kailan siya magte-terminate? Okay? So, elongation of RNA continues until a termination signal is reached. Okay? Ano ba yung mga termination signal? Alright? The raw independent, no, it requires formation of a hair loop. Okay? Raw dependent requires raw protein to release RNA from DNA. Okay, so take a look at this. So, ito yung tinatawag na hairpin loop. Okay? Once yung transcription, okay, yung RNA molecule nag-form na into hairpin, ibig sabihin, nakasalubong na yung termination signal. And RNA synthesis is about to end. Okay? So, yun lang yung tatandaan ninyo. Hairpin loop, pag nakasalubong yung hairpin loop sa raw independent, pero pag sa raw dependent, raw protein naman. No, it requires raw protein to release RNA from the DNA. So, ganun lang, no? So, initiation, elongation, termination. Okay? So, tandaan, sa initiation, kailangan merong DNA promoter sequence. Right? Para makapag-start si RNA polymerase to code for the RNA molecule. Okay? And for the termination, kailangan may makasalubong din siya or may magawa siyang hairpin loop structure, no? And yun yung palatandaan ng RNA polymerase to stop RNA synthesis. Okay? Alright. So, ano to? Ano ba yung product ng transcription? Okay? Kung yung DNA replication, ang product niya is another set of identical DNA molecule, si transcription, ang product niya is mRNA. Okay? mRNA. Sige. Si mRNA, no? So, tingnan nyo muna to, no? So, ito yung kinode natin for transcription. Okay? So, itong nasa taas, ito yung DNA molecule natin na kinode. Okay? So, yan yung kinode. Tapos, ito ngayon yung mRNA, product ng transcription. Okay? Since, kita nyo, single strand na lang siya because it is an RNA molecule compared to DNA molecule na double-stranded. Okay? Sige. Ang isang mRNA molecule, or we can as well call it primary transcript, contains several segments. Okay? So we have here the exon 1, exon 2, exon 3, and exon 4. Itong mga sky blue in color na yan, no? Those are exons. Itong mga green, ang tawag sa kanila introns. Okay? Introns yung mga yan. Alright? Si introns, no, are non-functional and they need to be spliced. Okay? Kailangan pong ma-splice ng mga introns. Okay? Kailangan hindi sila masama. So, ang isasama lang sa mature mRNA are the exons. Okay? Because they are the ones who are being used for expression of protein. Okay? So, ika-cut mo, meron tayong tinatawag na capping, splicing, and polyadenylation. Ang tawag natin dito is post-transcriptional modification. Okay? So, yung primary transcript, it's not final product yet, ha? So, kailangan pang matanggal yung mga molecules na non-functional such as the introns. So, it needs to be spliced first. Okay? Kapag na-splice na yung mga introns, meron na tayong mature mRNA product here. Okay? This mature mRNA will be used for the translation process later. Okay? So, okay. Tingnan natin to, no? So, mayroon tayong tinatawag na post-transcriptional modification. Post-transcriptional after transcription. Okay? So, yung initial product ng transcription or RNA synthesis will undergo certain modification. Okay? Isang process of post-transcriptional modification is RNA splicing. Okay? So, again, exons are coding sequences, introns are non-coding. Sa primary transcript, meron pang kasamang introns. Okay? But we need the coding sequence kasi magta-translate na tayo into protein. So, kailangan mga functional lang. So, we now have to splice the introns. Okay? So, tawag natin sa process is RNA splicing. Introns are removed and exons are spliced together. Okay? So, ito yung primary transcript, no? Meron siyang exon 1, intron 1, exon 2, intron 2. Okay? So, these are now, sabi natin, kailangan ma-splice, kailangan matanggal yung introns, kailangan exon lang, no? So, ito na ngayon yung final messenger RNA natin. So, aside from RNA splicing, what are the other process that can modify an mRNA? Okay? Pwede tayong mag-add ng 7-methyl guanosine cap, addition of poly-A tail, addition of CC-A tail, methylation, reduction, deamination, and rearrangement of glycosidic bonds to create unusual bases. Okay? So, yun yung pwedeng gawin para ma-finalize yung product ng transcription. Okay? Right? So, other process is we call alternative splicing. Ano namang ginagawa sa alternative splicing? So, cutting and pasting the exons in different patterns. Okay? So, it giving us more option. Okay? Sa RNA splicing, magkakadikit lang si exon 1, exon 2, exon 3, and exon 4. For alternative splicing, we can shuffle. Okay? And we can produce more RNA products. No? So, we can have here for protein A, exon 1, exon 2, exon 3. For protein B, exon 1, exon 2, exon 4. Okay, so we can do some shuffling. Alright. So this is now the after after the post-transcriptional modification, no, which undergoes splicing, no, RNA splicing, alternative splicing, capping, and all methylation, no, we now have mature mRNA. Okay, mature mRNA, which is ready for the next process, which is translation. So this mRNA will now transfer, no, will now use the information from the DNA molecule, no, to be expressed as a protein. All right. So that's all about transcription. So from our substrate, no, is a DNA molecule. It will undergo transcription. We will have mRNA molecule as a final product. Okay? So kapag sinabing transcription, it is as well called RNA synthesis. So DNA to RNA. Okay? So understand or remember the important enzymes, the RNA polymerase. Okay? So ano pa yung mga kailangang tandaan sa transcription? The DNA promoter sequence and the termination signal. Okay? Kailan sila mag-start? Okay? Alright, so if you have any questions, feel free to message me.